Good morning, everyone. My name is Jielun Hu, and I come from Guao Food Research Center, and my supervisor is Dr. Joshua Gong. And today I will talk about the selected Lactobacillus isolates attenuates Salmonella infection by modulating the gene expression of Salmonella virulence factors and cytokines in chickens. This is the experiment design. We used the 1-day-old chicken as the animal model, and we used the Salmonella to give the infection to the chicken. And the concentration is the 10 to 4 colony foaming unit per chicken. And we used some Lactobacillus treatment. The concentration is 10 to 6 to 10 to 7 colony foaming unit per chicken, and there are 4 treatment groups, that is low dose LB1 and high dose LB1. LB1 is a kind of lactobacillus from the chicken, and also we have the mixture of 4 lactobacillus, 3 from the pigs and 1 from the chicken, and also with the low dose and high dose mixture treatment groups. And we want to talk about the defense of the lactobacillus effects on the chickens into 2 ways. The first is from the Salmonella itself. We want to evaluate the SPI1, that means the Salmonella pathogenicity island 1, which is responsible for the gut infection virus gene expression. And also we want to look into the host mechanism. That's the way we use from the examine the cytokine gene expression, which related to the immune response. And this is the sample that we obtained. For the virulence gene expression of the Salmonella, we used first we use the Salmonella to give the chicken infection, and also we give the Lactobacillus treatment. And after 1 day post-infection and 3 days post-infection, we collect the cecal digester of the chicken, and then we do data analysis. But for the immune response analysis, we only collect the cecal, tonsils, ileum, and spleen for the cytokine expression analysis. And we also collect the samples for the 1 day post-infection and 3 days post-infection. Especially for the cecal, tonsil, and ileum, we want to look into the effects of the lactobacillus on the local immune organs. But for the spleen, that is the systematic immune organs that we want to look into. That's the results. Firstly, we found that the Lactobacillus treatment did not reduce Salmonella counts in cecal digester, but it attenuated infection to the chicken liver and the spleen. And then we found that the effects of Lactobacillus on Salmonella itself, virulence genes, that means SPI1 virulence gene, interestingly, we found that 10 genes has been downregulated by the Lactobacillus treatment. And for the effects of the Lactobacillus on the chicken immune response related genes, we found that the pro-inflammatory cytokine interleukin-6 from the local organ cecal, tonsil, ileum to the systematic immune organ spleen, the interleukin-6 has all been downregulated by the Lactobacillus treatment. And for the Th1 cytokine, interferon gamma in cecal, tonsil, and interleukin-12 in ileum and spleen, it has been upregulated by the Lactobacillus treatment. But for the Th2 cytokine, interleukin-4 from the local ileum to the systematic organ spleen, it has been upregulated due to the Lactobacillus treatment. So finally, we got the conclusion, and we found that the tested Lactobacillus isolates can attenuate the Salmonella infection to chickens by 2 ways. The first is to reducing the Salmonella SPI1 virus gene expression in the chicken cecal, and the second, it can altering the pro-inflammatory and the Th1 and the Th2 cytokine gene expression in chicken immune organs. That's all my talk. Thank you very much.